Our bodies are made up of trillions of cells, and each cell contains a complete set of instructions that dictate how we look and function. These instructions are encoded within our DNA, a long, spiraling molecule that resembles a twisted ladder. The rungs of this ladder are made up of 4 chemical bases: adenine, thymine, guanine, and cytosine. These bases are paired together in specific combinations, adenine with thymine and cytosine with guanine, to form the genetic code. Genes are specific segments of DNA that carry the instructions for building proteins, the workhorses of our cells. They determine our hair color, eye color, height, and countless other traits. Our genes are like a vast library of information, passed down from generation to generation. However, our genes are not our destiny. While the DNA sequence itself remains largely unchanged throughout our lives, the way these genes are expressed can be influenced by factors beyond our genes. This is where epigenetics comes into play. Epigenetics is a relatively new and exciting field of science that explores how our environment and lifestyle can alter gene expression without changing the underlying DNA sequence. Imagine your DNA as a book. The words in the book represent your genes. Epigenetics is like a series of bookmarks and highlighting marks that tell your cells which pages to read or genes to express and which ones to skip over. These marks, known as epigenetic marks, can be influenced by a variety of factors, including diet, stress, exposure to toxins, and even social interactions. Unlike genetic mutations, which are permanent changes to the DNA sequence, epigenetic changes are reversible. This means that we have the potential to influence how our genes are expressed through our lifestyle choices. Identical twins provide a fascinating window into the interplay between genetics and epigenetics. Since identical twins share the same DNA sequence, any differences in their appearance, health, or behavior can be attributed to environmental and lifestyle factors. Studies have shown that identical twins raised apart often exhibit differences in disease susceptibility, personality traits, and even physical characteristics. For example, one twin may develop heart disease while the other remains healthy, or one twin may be more prone to anxiety or depression. These differences highlight the profound impact that our environment and experiences can have on our gene expression. Section 4. The Instruction Manual: How Epigenetic Marks Work. Epigenetic marks function like an instruction manual that sits on top of our DNA, telling our cells which genes to activate and which to keep silent. These marks can be added or removed throughout our lives, influencing how our genes are expressed. Two of the most well-studied epigenetic mechanisms are DNA methylation and histone modification. DNA methylation involves attaching a methyl group, a simple chemical tag, to specific regions of DNA. This typically acts as a switch, turning the gene off. Histone modification, on the other hand, involves altering the proteins around which DNA is wrapped. These histones act like spools, and modifications to them can either loosen or tighten the DNA coil, making the genes more or less accessible for expression. Section 5. A Mother's Legacy, Epigenetics and Fetal Development. The prenatal environment plays a crucial role in shaping the epigenome of a developing fetus. A mother's diet, stress levels, and exposure to environmental toxins can all leave lasting epigenetic marks on her offspring. For example, studies have shown that maternal malnutrition during pregnancy can increase the risk of obesity, heart disease, and diabetes in the child later in life. This is because the fetus's genes are being programmed in an environment where resources are scarce, leading to adaptations that may be beneficial in utero but detrimental in a nutrient-rich environment. Similarly, maternal stress during pregnancy has been linked to an increased risk of anxiety and depression in children.